Today's spotlight in the Legacy video series is on the role of fatty acids in enhancing reproductive performance of dairy cattle. For many decades, the role of fat supplements fed to dairy cows was confined almost exclusively to its energy contribution and effects on milk yield. In more recent years, attention began to shift from fat as just an energy source to fatty acids as regulators of cell function and metabolism. Included among the tissue effects of fatty acids were reports of improved reproductive performance. I'm pleased to have with us today one of the world's leading experts in the area of animal reproduction, Dr. Bill Thatcher from the University of Florida. Dr. Thatcher is among one of the most highly cited experts in reproductive physiology of dairy cattle and has received numerous awards for his accomplishments. Among his many areas of research in reproduction of dairy cattle was his interest in examining the role of lipids in modifying reproductive physiology. Dr. Thatcher played a key role in establishing links between intake of specific fatty acids by cows and their effects on reproductive performance. So it's with great pleasure to have Dr. Thatcher with us today to explain the history of events that led to our current understanding of lipids in reproduction. Bill, thanks very much for being with us today, taking time out of your busy schedule.
Thanks, Tom. It's a real pleasure for me to be here, and I really appreciate the opportunity to review the work that we've done over the years, collaborative efforts among my colleagues and students and allied industries, and I appreciate the allied industries cooperating with universities. Is really critical in development in this area.
And it's sure that cooperative effort that we want to have come out here today. But before we get into the science of reproduction and fatty acids, let's step back in time and take a look at where it all began for Dr. Thatcher. Where were you born and raised?
I was born in Baltimore, Maryland, and had a very interesting background. My mom was raised on a dairy farm. My grandfather had a dairy farm up in Falston, Maryland. And my father was a city boy, and he met my mother when she was in nurses training. And first slide that's up here shows my early attachments with livestock, etc. But at the first slide on my little rocking horse, kind of destined me for kind of being involved in animal husbandry. And the pictures below that are my cousins, and we go up to my grandfather's dairy farm every Sunday. It was like paradise up there. It was, we were all about the same age. You know, it was just during and near the end of the war, and we're just really good friends. And my grandfather's dairy farm was just a treasure house of activities that we grew up in.
How far away was the farm from where you grew up?
Probably about 20 miles.
Okay. So you could get there easy enough and often?
Every Sunday.
Yeah, okay.
For a big family dinner.
Yeah.
And then I was about 12 years old, we moved into the countryside, which is actually suburbs now, but you can see with me on my horse Dawn, and there's a little colt there that's actually suckling the mammary gland. And I was just fascinated with that. And when it came time to go to college, I majored in animal husbandry at the University of Maryland, and really enjoyed my undergraduate degree. And probably when I was about my junior year, I started working part-time. I think all of us worked in those days, but it worked part-time out at the USDA at Beltsville, Maryland, real close to College Park.
Yes.
And I was able to really have a unique environment with some unique mentors. And the next slide shows two of my mentors that were very important. Charlie Kitty was at the USDA, the University of Maryland, and I graduated with my BS degree. He said, why don't you do a master's degree? We were doing a lot of projects in the laboratory as an undergraduate. I was kind of a grunt, but I learned an awful lot. And so he got me set up for a joint assistantship with the University of Maryland. And Walt Williams, a reproductive physiologist, was on my committee. And The gentleman on the left is Alan Tucker, and I went to Michigan State University, and these were two extraordinary mentors that I had. And both locations, we had a kind of an interdisciplinary type of environment, and it was really very, very, very important for me in my thinking and ability to cooperate with other people, and being mentored in the scientific method, and during my PhD with Alan Tucker, the importance of publication and rigorous use of the scientific method and application of statistics, and it was just really a very important environment.
Yeah, the contributions of Dr. Tucker to our industry has been phenomenal.
Yes,
for sure.
it has. And he has a legacy of students. And
yes.
I think I was his third student that he had. Yeah. I put in the next slide, I asked, when I was asked to talk here, I was trying to think back when I first realized or started questioning the importance of lipids. And I think I was my senior year in college, and Harold Hawk and Truett Brinsfield at the USDA, they were looking at the endometrium in progesterone-treated animals. And you'll see on the left, it's at day 4 of the cycle. And the clear area, it represents the uterine lumen, and then the epithelial cells line both sides of the lumen. And probably about day 9 or so of the cycle after progesterone exposure, and they actually injected animals, you'll see these little black dots, and those are lipid droplets. And I saw photomicrographs where they had going into the embryo, and it was really something that captured my imagination at the time, and I remember very clearly. And that was back in probably the paper was published probably in 71 or 72, but that work was being done when I was still at Beltsville,
okay.
Before I
so
went
you were at the University of Maryland,
yeah.
working on your master's degree.
Right. Exactly.
When that work became
right.
Yeah.
The first use of isotopes, Walt Williams was using labeled progesterone, and you could see all these changes in the uterus, and that really stayed with me a lot.
Yeah.
See, the next slide are some mentors. This is when I first went to the University of Florida. Fuller Baser is the one on the left. We were the two reproductive physiologists that went to the University of Florida within 6 months of each other, and we networked in with Dr. Warnick, who was a long-term person there. And then the gentleman in the middle is Dr. Donald Barron, and he worked with Sir Joseph Barcroft at Cambridge University, and then went back to Yale University after the war, and then retired and took an endowed chair at Florida. He was a real mentor to the group at the University of Florida, and was just really instrumental. I gave a seminar on my PhD just after he arrived, and he took me aside after it, and he said, "I have something I want to show you." He goes to Austria, in Vienna, every summer, and he came back and he gave me this paper published by obstetrician gynecologist in probably about 1875, and he had programmed the mammary development in humans with the uterine development from the embryo through puberty, through the estrous cycle, all through pregnancy, suckled, non-suckled mothers, up to the time through premenopausal, postmenopausal, and linked the mammary gland with the uterus. And it was really a very perceptive talk, because my mentor, Alan Tucker, was doing that with DNA and RNA with both the cow, different physiological periods, as well as the rat. So that was a real important environment.
Now, just to back up for a second, did you move right away from your PhD at Michigan State to University of Florida?
Yes.
Okay.
Yes, in 1969, January 1.
Okay.
is when we arrived.
Yeah.
The other important issue that kind of deals with the lipids was Jim Lauderdale had shown that prostaglandin F2 alpha was luteolytic and became a pharmaceutical product for being able to synchronize animals. And here's just a slide showing an injection of prostaglandin, which just regressed the corpus luteum. This is one of my students, John Chenault, did this, and you see a drop in progesterone and a transitional rise in estrogen and an LH surge. And to see something so powerful, we did a lot of work with prostaglandin F2 alpha physiologically. and realizing that that's synthesized from arachidonic acid, and that it must be a very important physiological phenomena that's going on in the uterus. And we were interested in really studying that. And so that was a area that we got into. And how we developed that kind of shows in the next slide. If you just look at the picture in the top there, you see an ovoid embryo. That's probably about day 14 after breeding. And you can see the arrow to the right is a long filamentous embryo that you could see at day 15, day 16 in the cow. And so that tremendous expansion of the membranes represents a lot of proliferation and a lot of need for fats.
For nutrients.
Nutrients,
lipids.
fats, and lipids for the membranes, and stimulation of proliferation. So we first then started looking at how much prostaglandins are produced by the blastocysts. And Greg Lewis, working with us at the time on a postdoc, quantified PGF2 alpha, PGE2, and the metabolite of PGF2 alpha. And you can see on the quantity that's produced by the blastocysts between day 16 and day 19 goes from 2 up to 22 micrograms per blastocyst.
Really jumps up.
It really jumps up, and so does PGE2. And these were done in culture and over a 24-hour period. And we went for 24 hours because we had done some isotopic incorporation of arachidonic acid and getting it into the cells, into the tissues, and getting the pools labeled. 24 hours was a period of time that we needed to do that. And so that was really a pivotal study for us, and it's refreshing to see where that's gone to up until today in terms of other sorts of prostaglandins. And the next slide really kind of shows a model that we had used. You're looking at a cell membrane there, and there's the oxytocin receptor, and oxytocin binds to the receptor. It activates a PLA2, a phospholipase, and the arachidonic acid then is converted by PGH synthetase to products. And you see there's PGF2 alpha, PGH2, and you have a series of products there, PGF2 alpha, PGI2. You can see that there's PG2 series, PGE2, and this PGD2 is metabolized and actually is a candidate that binds to very specific PPAR receptors in the membrane of the embryo. So this is all kind of developing, and I tried to capture where our diet might fit here. Is if we supply through the diet polyunsaturated fatty acids, they also are taken up into those membranes and can be mobilized, and so we can get a competition between, say, linoleic and arachidonic acid, linoleic being converted to arachidonic acid. But if you gave like a fish oil polyunsaturated EPA icosapentaenoic, it'll compete with arachidonic acid for conversion, and we can regulate the amounts of PGF2 alpha and PGE2 that are produced.
Yeah, I'm sure there's lots of interactions that are going on between these individual fatty acids.
Exactly, and it's
pathway.
And at this stage, we've done the studies. It's a kind of a competition, not so much a direct gene regulation of PGH2. It's just availability of substrate that was competing. Whereas the embryo produces a protein that will actually decrease the PGH2 synthetase. So the two of them could have additive effects. So as we're trying to increase the survival of embryos, and we want to reduce prostaglandin availability for CL regression, we can play on that in terms of getting kind of additive effects. So that's a little bit into our thinking. And then the next slide shows a experiment that we published in 2011 with using microarray, looking at the transcriptome in the endometrium, and we asked the question: is there a difference between a cyclic animal and a pregnant animal in terms of what's being transcribed in the endometrium? And then also, is there a difference between lactating and non-lactating animals? And so we had first calf heifers, and we either milked them or or did not milk them, and then we went out, synchronized them, and either inseminated them or not inseminated them, and recovered the embryos. And we looked at the genes. And I just want to take one area, and it's the gene expression of fatty acid-related genes. You can see the list there: fatty acid binding protein, fatty acid synthetase, a series of genes that are influenced by pregnancy. The two columns on the right is a cyclic pregnant, these are in red, and every one of those genes you can see is upregulated at the same stage after ovulation at 17 days in the pregnant animal. So the pregnant animal has a very important difference in transcription of these genes that are involved in fatty acid transport, synthesis, and in terms of lipid biosynthesis in the endometrium. So I think that's a very important thing that's going on that we can play with in terms of being able to.
And that's still developing. I mean, we still have a lot to learn about it, but this certainly was important information and the basic understanding of that.
Exactly.
Yeah.
And then the next thing that we were interested in, Tom, was trying to transmit this into the whole animal in terms of reproductive management.
Yes.
So one of the questions we asked, and it was a debatable issue in goats, when you have parturition, you have a large increase in prostaglandins for the first 14 days. And there was some controversy as to whether this was coming from the mammary gland or whether it was coming from the uterus. So we did a series of experiments. One of them was just to do classical hysterectomy right at parturition. This involved cooperation with our Martin Dross in the vet school. We just prolapsed the uterus and basically amputated it and then measured prostaglandin, and it just completely disappeared. So and then we cultured like the caruncular tissue and showed that the uterus was a major player here of postpartum. We asked some questions: is it involved in uterine involution and things like this? Calculated the production rate. And then the next important part is, can these fatty acids regulate prostaglandin? And so we did culture studies with stimulating with 4-ball esters the prostaglandin synthetase system, and then we looked at the effects of alpha-linolenic, EPA, and DHA on the biosynthesis of prostaglandin, and we could show that it competes with arachidonic acid, and we could actually reduce the production. That was a very important thing. And then I think one of the first in vivo experiments we did, this is a little bit of naiveness on our part, but it was very revealing. Matt Lucy was in the laboratory at the time, and we took heifers and synchronized them, and then from about 9 to 14 days of the cycle, we had gotten intralipid from the medical school pharmacy and actually depleted their supply of shoes for intravenous infusions for human patients. And we infused 1 liter per day for a 4-hour period. And then we measured prostaglandins at the end of the infusion period and estradiol and follicles. And we were shocked. We got an immediate increase in prostaglandin in the peripheral blood measured by the metabolite. Follicles were getting larger, and we were getting increases in estradiol, and it was much too quick. I wasn't sure it was really maybe just the effect of the infusion causing us kind of a reaction to it wasn't pure linoleic acid. It had a series of fatty acids. And the other issue was, though, that it was influencing the follicle, small numbers of animals, but it was really a pretty revealing type of experiment. And then after that, we went on and did our first calcium salt experiment.
Okay.
And what we did was a pretty simplistic experiment, but it was a lot of numbers. And basically, we took postpartum cows, and probably at about 25 days, we gave them prostaglandin to regress the CL, so they all don't have a CL on the ovary, and we put in what we call a cedar device, an intravaginal device, just to kind of hold the cows so they wouldn't ovulate. And then we kept it in there for about 12 days, and then pulled it. And then these cows were on calcium salts of palm oil. This would be a Megalac product at the time. And they were on that through this whole period with controls. And we could see very clearly that we were stimulating follicle size. And we were holding the follicles, and they were getting bigger and bigger and bigger. And that was a pretty important experiment for us. If you look at some of the first things that happen now in postpartum cows, when you supplement them with these calcium bypass fats, you'll see earlier returns to estrus, and we're stimulating ovarian follicle development. So that was a really interesting set of experiments for us in terms of knowing what's going on.
Yeah. Now you must have had the question somewhere along in there: is it an energy response or is it a lipid response?
Yes, that's
responsible for that.
That's right. We did other experiments where we looked at the effects of supplemental calcium salts. We had isocaloric diets. And then we had diets without the calcium salt as well. It was not isocaloric.
Yeah.
And we could show that there's effects. We even had a BST factored in there, and we could see that there's very, very clear effects of the fatty acid. It's not just an energy issue. But you raised a very good point, because we did look at an experiment in a factorial where we fed a high degradable protein and a low degradable protein diets with and without calcium salts. And on the high degradable protein, we had lost a lot of body weight and body condition. And by supplementing with the calcium salts, we were able to counter that and get them up to the control levels that were on a low degradable protein diet and with or without calcium. So there is an energy issue there as well. And I think as we see the effects of these and some of the molecular control and some of the things that are regulating, that it's more than just energy. We can capitalize on these different things, different types of fatty acids, which is what I want to get into now on a farm, at the farm level.
Yeah.
And so the next slide is for me is a very important slide. With this basic knowledge, we were trying then to implement things at the farm level, and I want to acknowledge two of my colleagues, Jose Santos on the left and Charlie Staples on the right, and we all teamed up, and there's just a large number of students have contributed to these various projects, and our dairy producers that we were able to interact with and with allied industries. And it takes a team approach. There's a veterinary component too that's involved in all this, and that's what it really takes. And collectively, in other areas that we work on, we kind of develop systems that really control the time of ovulation, and we can get good fertility, and we can start asking the question, does these fats have any beneficial effect on milk production, the ability of animals to get pregnant, ability of animals to maintain their embryos. Yeah. So that's kind of what I wanted to share with you, just a couple examples. And this next slide shows a very important experiment that we had done. This is Flavio Silvestri, part of his doctoral program. And we were looking in a 2x2 factorial, feeding a certain dietary regimen for the transition period from 27 days before parturition to 30 days after parturition. And you see we're feeding calcium salts of palm oil, which is enriched in 16-0. And then we were comparing that to cows, a large number of animals here, that are fed safflower oil, which is a good source of linoleic acid, 18-2. And we have a large number of animals there, you can see. And our thinking here with Tom was to be able to feed a diet rich in linoleic acid that would have pro-inflammatory. We were trying to boost the immune competence of these animals, and we looked at that. And
well,
then,
you're beginning to try to sort through individual fatty acids.
Yes.
And
yes.
Because you've established that it was fat, not energy, and now what types of fat?
Yes.
Yeah.
Yes. And so we picked the safflower oil, for example, as a pro-inflammatory fatty acid, and trying to boost the immune capability of the cow in the postpartum period. And then the 31 days, we call it the breeding period, out to 160 days, we fed palm oil versus calcium salts of fish oil. EPA and DHA. And our thinking there is that we wanted to have an anti-inflammatory diet. Cows that have infections, for example, they can carry some of that over into the breeding period in terms of subclinical endometritis. And if we can just kind of reduce the inflammation at those stages, that might have a benefit. And there might be some very specific effects as well on the endometrium as well as the developing embryo. So that was our thinking there. And the next slide just shows you that our hypothesis relative to the pro-inflammatory and anti-inflammatory is correctly. This is looking at TNF-alpha in the top. These are actual cells, peripheral blood cells, white blood cells, neutrophils specifically, that were collected from these subgroup of these cows. And we stimulated them with lipopolysaccharide, and you can see in the middle, 2 bars in the middle on the upper graph, we stimulated TNF-alpha, which is a cytokine that can stimulate acute phase proteins in the liver. And you can see the red is the diets that were on the sapphire oil, or they had gotten linoleic acid exposure more than the controls. And you see there's a stimulation of the cytokine TNF-alpha. And then out at 80 days in the lower right is when the animals were on the fish oil, calcium
acids.
Salts, right.
Yeah.
And you can see in the middle 2 bars there with the lipopolysaccharide that those cells, neutrophils, were actually had a decrease in TNF-alpha. So, and we measured a bunch of other things. We measured haptoglobin and other cytokines, and it's consistent with that we're able to regulate the immune competence of these cells with these markers.
Yeah, the results are quite dramatic in that study,
yes.
And clearly different between individual groups of fatty acids.
That's right, exactly.
Yeah.
And then in the next slide shows the pregnancy response. This is the day 60, and you'll see it says saturated. It's a 2x2 treatment factorial that you have there. Then you have an N6 saturated. So like an N6 saturated would be a group that had gotten the sapphire oil in the transition period, followed by palm oil in the breeding period. And so the 2 groups on the right in the first figure, the bar graph, pregnancy at day 60, they had gotten the fish oil in the breeding period, preceded either by high sapphire oil, linoleic, or by palm oil. And they averaged about 52% fertility, and was about 6% higher compared to the average of the 2 groups that did not get sapphire oil in the breeding period. And then on the right is the pregnancy loss. The animals, what percent of the animals lost their pregnancy between 32 to 60 days, and that was reduced from about 12% down to about 5%. And these differences are significant. We had a lot of numbers of cows in these things. So this was a indication of a pregnancy effect. There's several papers now published in the literature. There's one just came out this past year or so in Journal of Animal Science from investigators in Iran. They were trained in the States and back in Iran doing some very nice work. Helen Petit in Canada is associated with that paper. And they're seeing exactly the same thing, which is comforting. And there's other sources, too, that are either in progress or done in some other laboratories that are showing these types of advantages. And so I think this is pretty important. You notice that, and if I have the next slide, in this experiment that I just shared with you, we're having to change the diets. So we've done a series of experiments with Dr. Santos and Dr. Staples and the groups where we're trying to get down to where we can feed one diet. And so this is an experiment that was recently done and in cooperation actually with Virtus Nutrition, where we tried to change the ratio of linoleic acid or N6 fatty acids to N3 fatty acids. And so looking at our ratios, 6-to-1, 5-to-1, 4-to-1, and this particular experiment, we started at 6 to 10 days in milk, and we're going to be doing starting it earlier in the transition period, but this particular experiment was started at the 14 days in milk and continued out through 106 days, 13 weeks. And if we look at the next slide here, we also did an intermammary challenge with LPS to see if there was any types of effect. And I just want to highlight a couple of responses. First one, next slide shows the milk production response. And here you can see what we call R4 is the top line. So the ratio of omega-6 to omega-3 of 4 stimulated milk production. And you can see it kind of steps down to R5 and then down to R6. And you know, a lot of our experience, if we start to see really substantial or increases in milk production, this represents, in my opinion, a more healthy, productive cow,
Yeah.
and they're more fertile.
Yeah.
And so I think this was very, very encouraging.
And just to clarify for our viewers, Bill, R6 is more similar to a typical TMR without any supplemented omega-3s. R6 is. And then as you supplement with additional omega-3s, you go from R6 to R4.
That's right.
Yeah.
And R6 is a ratio. It's I think a lot of the rations that are out there were probably in the N6 to N3 ratio of 12.
Yeah.
So
higher.
Even higher. And in the humans, it's 14, 16, something like this, you know. So this is a interesting type of response. And one of the questions is what's going on in the uterus. So the next slide shows the actual arachidonic acid and the actual EPA concentrations measured as a percent of the total fatty acids. And you can see the blue line, as you go from R4 to R6, you can see there's an increase in arachidonic acid. And the amount of EPA going from R4 to R6 is declining. So feeding the lower ratio, 4 to 1, which would have more of the EPA omega-3s, you get a decrease in the concentration. So this is being absorbed. Not everything's being biohydrogenated here, and we've looked at this in a variety of other tissues as well, and you know, we can capture increases in the concentration.
That shows a highly significant increase in the endometrial tissues
Right.
as a result of supplementing with the omega-3. So like you say, they are getting past the rumen microbial transformations and finding their way into the tissues.
Yes, we're.
Yeah.
And we've looked at this in numerous experiments. But the next slide shows just some marker enzymes in the endometrium that we associate with regulating prostaglandin secretion. And if you look at that, for example, the first two pairs are estrogen receptor. Then there's the next three columns is the oxytocin receptor, and then PGS synthetase, and the last one is the PGE2 synthetase. And in all of those cases, you can see when we go up to R6, we're stimulating estrogen receptors, we're stimulating oxytocin receptors, we're stimulating prostaglandin synthetase and PGE synthetase. And these were done by biopsy of the endometrium at 9 days after ovulation. So the diet is influencing that. Or conversely, if you're feeding a low ratio of N6 to N3, and that's how we marked this. We looked at the data two different ways, but low, medium, or high concentrations categorized, or if we had done it by the R4, 5, and 6 ratios, you get the same type of response. But conversely, we're decreasing those things that are leading to a synthesis or an increase in prostaglandins. We don't want to wipe out prostaglandin production completely, because it's very important for differentiation of the trophectoderm, and we have data now to indicate that there's PPAR receptors, particularly PPAR gamma, in the membranes of the embryo, and some of these products are binding to binding to that, they hybridimerize and are very important for the function of the embryo. So in ending here, I just want to make a point, and the next slide kind of captures the national herd. This is the phenotypic trend, and this is a summary from the Animal Improvement Laboratory. And you can see then the red line that daughter pregnancy rate is going down, and you from the 1975 up through 2011, you can see that descending part of the curve, and then in the mid-80s, we're starting to see the phenotypic trend in daughter pregnancy rate rising. So such things as these nutritional management factors, how we manage dairy cows, more comfort, heat abatement, proper dietary management, these synchronization programs, we're getting more animals bred earlier, and it's really very exciting. And you can see now it's increasing back up to the levels that we saw in the mid-1980s. Yep. And so I think this is really a very clear signal that we're kind of reversing the phenotypic trend, and if you look at the genetic trend, and I didn't show that here, it's starting to rise very slowly. So as we started selecting for productive life and daughter pregnancy rate, and I think now we're going to have sire conception rate. Now we have the genomics to be able to predict the female in terms of which ones are going to be more fertile, and we can start selecting both on the female side as well as the male. We can make a lot more progress. So it's exciting times, and I'm trying to share with you today that, excuse me, that the nutritional component with the fatty acids is a very important part of this. The more we learn how to be able to understand the mechanisms and the timing and make it more practical for dairy producers to use, I think this is going to be a very important contribution to our dairy industry. Yeah,
well, you've made it very, very clear the linkages that exist between fatty acid nutrition and reproductive physiology and the hormonal development and so forth. And that's a very, very that's why we're here, because it's a very important breakthrough in lipid nutrition of dairy cows, to realize what fatty acids have to offer in the whole scheme of manipulating and controlling reproductive performance of animals. And the University of Florida, I think, has been very, very unique because of the collaborative effort, the very good collaborative effort you've had across the number of disciplines to be able to pull all this together. That's
very, very important, and I think that's very insightful for universities to recognize that this is what it really takes. And we're interacting with the people in the medical school. We interact with you. You've been very helpful on our fatty acid analyses, and it really takes a team effort. And so we're really excited about that. And I haven't talked about carryover effects potentially of some of these diets on the newborn calf. So the mother's being fed this in the transition period, and we have some evidence that these calves, females that are born from these types of nutritional manipulations, may actually be producing more milk in their first lactation. And so the epigenetic effects are really exciting.
Yeah, and that's an area that's just beginning to be recognized and emerging. And I hope we have you or somebody else back here one of these days to talk about all the great findings and research there is in that area, being able to control the maternal nutrition and have it carry over into the offspring.
We have a great crop of young scientists out there that are really doing excellent jobs, and we might thought of as mentoring them, but these students and young faculty and even seasoned faculty are mentoring us as well. So
very good.
Very good. Thank you for having me. Really appreciate this very much.
Well, thanks for sure for coming and visiting with us today and laying out the great history and thinking that went on in this important area. Thanks again, Bill.
Thank you.